So, welcome class. I hope you can see the screen of mine and you can hear me. Please confirm in the chat box if you are present and able to listen me and able to see my screen clearly and crystal, so that we can start based on that our first topic of today. Students, please confirm me that whether you can hear me properly and type in the chat box that if everything is fine. Okay, good. Mm, I will change the heading. It's from my university side, but I will send you the others presentations. So, what is the need for study of enzyme engineering or enzymology? We can also say why we have to, study this part. What is the reason behind it? Um, why does it came that the importance of mainly the enzymes started to play a role in the last 5 years? Earlier in our time, in 2005 when I did my BTech in biotechnology, we never had this subject of enzymology or enzyme engineering. Mainly we were studying about enzyme in our biochemistry subject, and that's it. But now, as the industry is growing, there is a lot of demands for the microbes and enzymatic catalyst that which could bring a lot of properties, lot of good things to produce really nice products and in very quick manner within limited time and very less expenditure also. So the input is very less in them while using biotechnological abled your enzymes such as various microbes we can use, and the output that we get is massive, it's huge. So in that sense, it's a must for our future. And if we have understood the basics about the enzyme engineering, that how the enzymes are working, what is the basics behind it, how the kinetics is working, it will make a strong background for our future studies related to all enzyme studies then. So let's start today with the first topic, that is the history. How, when, and the enzyme engineering is being started. So let's start. So basically, as we know that what is the role of enzyme actually? Enzyme is nothing. It came like a party. It's a in between party, which comes attached to the substrate and make the reaction fast and make product, and it's keep being used within 1 minute million times, and it's being never like finished in between them. It can be reused. It just helps to make reaction fast, catalyze the reaction fast, and from substrate and to production, it make that thing without any harm, any problem to it. It make the things very fast. That's the main role of our enzyme is. So enzymes, if we see the definitions, that enzymes are proteins that catalyze, that is increase the rate of chemical reactions. So they are basically helping us to fast the reactions, to make the reaction fast. That's why the enzyme engineering is there. Then what is enzyme engineering is? Enzyme engineering includes the applications of modifying an enzyme structure, modifying the catalytic activity of isolated enzyme, so that we can produce new metabolites, allow new pathways for reactions to occur, to convert from certain compounds to other. So let's say in biotransformations, in phytoremediation, or in the oil spills, all these environmental biotechnological environmental problems that we are facing at the moment, we can all clear them up with the help of your enzyme engineering. So we can use these techniques in order to solve our problems, because if we put some microbes which are eating your oil spills in the water, in the ocean, so they will finish them within few weeks, and we have clear crystal water at the end of the day. Yeah, same hold for the soil. We can have some enzymatic bacterias in the soil. If there is a lot of metal ions are there, so they will eat those metal ions and produce organic particles in the soil. So they are being very useful for all of us. So that's the various applications that we can use for. So some history of biotechnology. So basically, biotechnology is used for bread, beer, using yeast in Egypt part. So it's before Christ. So if we see from the perspective of enzyme engineering in the biotechnology, so we were already creating these breads, beer using the yeast in the past, and also in China, Sumerian Egypt, production of cheese and wine was also one of the prominent tradition that was going there. And some history of biotechnology with respect to first vaccination. So nowadays vaccination days is going on. So in 1797, Edward Jenner was an English scientist who pioneered your smallpox vaccines, and also known as father of immunology. So he created that part in order to enter in our body attenuated microbes or virus, so that we can produce some like you know some T lymphocytes, B lymphocytes in order to fight that virus. So this is what at the moment the Covishield that we are getting in India is based on also. So we had having a attenuated virus, a dead virus being injected into our body, and after seeing that, our body will start producing a lot of B lymphocytes and T lymphocytes. So our immunity will be increased against this virus. By the time we are infected with the real virus, then we will be having a lot of immune cells against that. They will fight for the same, which was not present in our body before our vaccine shot. So then Mendelian inheritance in 1865, who is also known as father of genetics. He's an Austrian Hungarian scientist, Augustinian priest. Was in a Catholic church. He was a priest, and in the background, in his backyard, he worked with a lot of peace experiments. You might all aware of. Um, and produces really good results in his times, various laws also, but they were not accepted as usual whenever a researcher is finding something. So they are not being appreciated at that time, but after few years the research is being carried out and being appreciated later on. Then in 1877, Edward Buchner, who got the first alcoholic respirations with cell-free extract he did. He's a German chemist and also got Nobel Prize in chemistry for his work on fermentation. So he's the first person who was dealing with the fermentation and got the Nobel Prize winner also in 1907 for the same. Then in 1894, lock and key model, Hermann Emil Fischer. He brought this into the enzyme world, that how your enzyme and substrate, they're acting like a lock and then key. So only the substrate will fit to only to that enzyme which has its shape, where it could attach to its catalytic substrate, but not all other substrate could be attached to enzyme. So it's like a key. Only one key will fit to a one particular lock. Not all keys could fit to one lock only. So they are very much specific about that, about the position. Only then the interaction will happen between your enzyme and substrate. And for this, the winner of the Nobel Prize winner was 1902 in chemistry. He also got the Nobel Prize winner in 1902 in chemistry for his thing. So Hermann Emil Fischer. You can see also their year of living from 1860 to 1970, from 1877 to 1947, till our independence of India. So remember, like these people, like from here 1749 to 1823, 1822 to 1884, he lived very less years, not so much. So these people, in their lifespan, when they were doing the things, they achieved very big things, which have brought them, like on today lecture in the morning that I'm teaching them, and I have these pictures of them respective. So they have concluded something really nice for the society. That's why they are in our textbook. That's why they are in our yeah, they got these prizes also, big prizes. It's just because they did something really unique for the society. So we should appreciate each scientist, each one in the past whosoever did a big discovery in order to make society a better and healthier and wealthier. Then in 1928, first discovery of antibiotic was done by Sir Alexander Fleming. From 1881 till 1955, he worked for the same. He was a Scottish biologist and pharmacologist, and he got for the discovery of the antibiotics in 1945 Nobel Prize winner in physiology of medicine. Yeah. So these Nobel Prize things are still going on in the society from 19 um, you know, we are having a lot of in 2020, I heard that one of the students was saying about that he was working on the CRISPR-Cas9. So in 2020, the prize for the CRISPR was given to a lady, a scientist who have discovered in 2016 about this CRISPR-Cas9, but she was appreciated after 4 years, and she got the Nobel Prize winner for finding the CRISPR-Cas9 technology, because that has created a really the inventions that are being found out, if they could change one era of research, if they could make the life of people much easier and able to think wiser, so that's the thing that will be playing role in the society. Yeah. Then sequence determination of insulin. So this famous Frederick Sanger, who was famous for finding the sequencing, DNA sequencing, Sanger sequencing we call it mostly, to identify. It was done in 1951, though it's let's say 70 years from now onwards, but that was a quite very big thing in order to find out the human genome. Because if we can able to sequence one DNA, so we are able to find out the whole genome, which was completed in 2003 actually. But afterwards we never see back. By 2003, there a lot of research has been done in order to find out many diseases. So his contribution, Frederick Sanger, is really immense. Is really immense. So he got two times Nobel laureates in chemistry, one 1958 and one in 1980. So these stories, though students, these stories, though they are meant, you know, uh, in the books we always talk about them, what they are, but really, if you think from the biography perspective, from all these scientists, if you are able to read their biographies, there will be immense amount of knowledge to learn from them, how a scientific mind works out. Yeah, how scientifically we have to think of. So in that sense, they are pretty good. Then in 1953, we know this famous Watson and Crick, James D. Watson and Francis Crick. So they proposed DNA structure. First of all, you can see they build this ball and stick model with the steel pipes and having this DNA double helix structures revolving around from the center. And they were jointly awarded this 1962 Nobel Prize winner for physiology or medicine. So yeah. Then in 1978, recombinant DNA by Cohen and Boer. By Cohen and Boer. So they discovered this rDNA technology, that is your genetic engineering that we are studying by the American genetists. So they developed the method of engineering techniques also, and yeah. So it was also quite helpful in the society, enabled to create transgenic plants, enabled to play around the genes of plants and in human genome also, in order for the better human things. And then comes in 1985 site-directed mutagenesis. So this was done by Canadian biochemist, British-born Canadian biochemist Michael Michelle Smith. So he established this site-directed mutagenesis and got the Nobel Prize winner in chemistry in 1993. So basically, in site-directed mutagenesis means if we having DNA sequence like A, T, C, G, T, T, A, P, C, C, G, G, A, T, 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 and what if we remove one DNA sequence, instead of A we bring out C, or in the amino acid sequence we have aline, isoleucine, cysteine, tryptophan. All these different amino acids are there in the long chain of polypeptide of amino acids in a protein. So we change one amino acid group. Let's say we remove from cysteine to serine with the help of this technique of site-directed mutagenesis. So this will produce a big change at the physiology of the function of the cell, and leads to the different functions in the animal also. So there are many mouse models are being created based on a side-directed mutagenesis, where we have we kill one mouse, we just switch off our one side of amino acids, and then we can see massive amount of difference. Mostly they are done in order to for the better health of ourself, of our mouse also. Then come in 1988, the biggest discovery, the PCR. I hope you are all aware of this PCR, Carrie B. Mullis, from 1944 till now. He's still alive. So American biochemist who developed this polymerase chain reactions, and he got also the Nobel Prize winner in the same year, 1993, in chemistry. So which has revolutionized in order to have a good amount of DNA, which was not possible earlier. So whenever we isolate DNA either from plant or from the animal cells, we always get in nanograms, and in order to do the successful experiments, we need at least in 1 milligram or so. So that was not possible earlier, but with the help of PCR, in which we just use thermal cycler and use three different temperatures: annealing, denaturation, annealing, and then elongation. Denaturation, elongation, yeah, like this: DAE, denaturation, annealing, and elongation, yeah. Based on three different temperature, we produce millions amount copies of our particular sequence of DNA that we are interested in. So this is also one of the biggest discovery, I must say, in the field of molecular and enzymology. Then comes something more prominent, that what happened in the enzyme engineering. So some definitions and terms that we should know about the catalyst. Here we have Oswald, who defines the term what is catalyst in 1893. Then in 1894, lock and key model was proposed by Fischer. Then in 1897, we demonstrated that enzymes do not require a cell. Then in 1926, enzyme is proved to be protein by Sumner. Then in 1958, induced fit model was proposed by Koshland. Then the first in 1963, first amino acid sequence of ribonuclease was reported. Then 1965, allosteric model of enzyme was proposed by Monod. Then immobilized enzymes were established by HFCS in 1970s. Then in 80s, protein engineering, chiral compounds, organic solvents, polymers. So advanced level of enzyme engineering started to happen from 80s. Then in 90s it went for the evolutions, and we can do also in 2000s computational designs of enzymes. So with the bioinformatics coming up, making up our structural structures we can predict with the help of computational softwares. This really has revolutionized the things. It has saved a lot of time. We do not need to synthesize these things in the organic lab, organic chemistry lab, and things are very easy to do so because of bioinformatics also. So some eight, so in total there were 8 Nobel Prize winners who got in the enzyme engineering from 1870 to 1988. Who did what? So let's see. Edward Buchner, he founded the first alcoholic respiration with cell-free extract. Then in 1893, Wilhelm Oswald, who defined the term catalyst. Then 1894, Emil Fischer, he found the lock and key concept. Then in 1926, J.M.B. Sumner, who found out the first enzyme crystallized urease from the jack beans, the enzyme urease actually. Then in 1951, Frederick Sanger and Hans Tupy, they sequence the insulin beta chain. Then in 63, Stanford Murray and William Stein, they produced this amino acid sequence of lysosomes, and ribonuclease elicited afterwards. Then in 1985, Michael Smith, who was a side-directed mutagenesis to change enzyme sequence. And then 1988, Carrie B. Mullis, he invented the PCR at the end. So all these people, Edward Buchner, Oswald, Fischer, Sumner, Sanger, Tupy, Murray, Stein, Smith, Mullis. So they did something really good in their field in the respective years, and that's why we are studying them today also. So some advantages and disadvantages of enzyme technology versus chemical technology. So first is that is a high degree of selectivity. These enzymes are quite selective, and when they start to work, they work in a particular manner. Environment friendly, they do not cause a lot of pollution at the end, they do not produce a lot of bioproducts in order to make environment bad. Beyond that, they're mainly based on the catalyzing reactions to make things fast. So and they are very less byproducts, not any many byproducts are being produced in this case, and they are non-toxic and non-flammable. All the reactions that are happening, they have no toxicity in that, and there's no flammable reagents are present. Then comes some advantages, disadvantages. Yeah, even though we can see some lot of cons in this, but they do cost a lot because you're protecting the environment, you're making things very fast, you're not having many byproducts, you're making your action fast, it's environment friendly, and it's very quite highly selective. Based on that, then you have to pay some amount for that too. So that's one thing. It's a bit not so stable sometimes, the things that we are working, your kinetics could go up and down, so we need to maintain the pressure, temperature, all the nutrients, everything need to be maintained in a way that the enzyme should work in a good way. Productivity is sometimes low. That's one of the disadvantage that's we have to deal with. But keeping the advantages part, which is overcomes these disadvantages, it's still promising technology which will be used for our future. Okay. So some nomenclature, that how the naming of these enzyme technology, enzymes are being done. So International Union of Biochemistry and Molecular Biology developed a nomenclature for enzymes, the EC number system. So in the EC number system, we have first number, the class of enzymes, second number, subclass of that substrate, then third number, subclass of the electron receptor, fourth number, serial number of enzyme found. So this is how you can see the six classes, top level of classification. This is the first number, that is oxireductase, transferases, hydrolases, lysases, isomerases, ligases. So there are many questions if somebody is preparing for exam. So which enzyme is doing which one and belongs to which category? So this is EC1 oxireductase. So what does oxireductase do mainly? It catalyzes the oxidation and reduction reactions. So all the oxidation reduction reactions are being taken care by oxireductase. Then transferases, this is as the name says, it transfer a functional group um, from one particular substrate to another. Then hydrolases, it's catalyzed various bonds with the help of hydrolases, your water. Then lysases, as the name suggests, it's cleave various bonds uh, other than the hydrolysis and oxidation. So it just cleave the bonds, not using your hydrolysis and oxidation process. And isomerase is like it's help in the catalyzing the changing the shape of a molecule within the molecule actually. So that's isomerization change within a single molecule, and ligases join two molecules with the help of covalent bonds. So you can check this link. So whatever things that we are studying students at the moment, so I will share with you over the Viber group. um, yeah, whenever we are finished with it, so I will share with you also. So industrial enzymes, um, so these are some of the important industrial enzymes being produced and have quite high importance in the society also. So these are the production scales which are of more than 100,000 level. So the product that we get is high fructose corn syrup, HFCS. And the enzyme that we use for the same is glucose isomerase. So this glucose isomerase is helping to produce high fructose corn syrup, which is being used, in your Bonvita, all these artificial products, Milo, all many things is being produced. So it's being used by many companies at the moment, and 100,000 lactose free milk. So it's being, this lactose free milk is produced by the enzyme lactase, because some are lactose intolerant in the milk. So in order to remove the lactose, we use this enzyme lactase to have a lactose free milk. Then acrylamide by nitrulase, cocoa butter by lipase, then aspartame by thermolysin, nicotinamide, nitrulase, ampicillin by penicillin amidase. So all these different products being produced by various companies and has quite highly importance to economy and also to the common people. So some important reactions, but we should not go into details with them. So that was enough. Like what were the various industrial enzyme presence, various classifications we have done. We have discussed the nomenclature. We have discussed why enzyme technology is important with respect to chemical technology, and we have discussed the various Nobel Prize winners so far given in the Society of Enzyme Engineering, and we have discussed a brief history of biotechnological famous Nobel Prize laureates who have produced very nice work to our society. So, and we have discussed today in the morning the first definitions of enzyme and enzyme engineering. What are the basic difference between them? So far we have done this part. So now let's continue further. So something about background of enzyme engineering. So we bring this term selectivity, which is quite important among others, which determine the usefulness of selectivity. So if our things are not particularly selective towards the things that we are working for, so it will be of no usefulness to us. So then these biocatalysts, your enzymes, so they need to be very selective, and organic chemists should pay more attention to the catalyst productivity, activity, and recycling. These are the key parameters for the application too. So that statement was given by E. Jacobson and M. Beller in 2004 about this selectivity, productivity, and biocatalyst. That they need to be, we need to pay attention over the recycling of the enzymes activity and catalyst productivity also, and also beyond that selectivity. So all these terms which I stated, that within them it's a whole chapters, whole books have been written on the enzyme activity, on the enzyme recycling, on the catalyst productivity, in order to make our industry be more, um, profitable. Actually, at the end of the day, when we are working for industry, we want profit, to get the maximum profit with the less input. So in order to understand that, we need to make sure that whatever the structure that we are working on, whatever the product that we are looking for, we should not know about each and everything about it from uh, top to bottom. So let's say this hydrolases enzyme industrial biocatalyst. So this was a new plant in USA, Geismar, which is having a capacity of 2.55 yeah, TA, and this is the enzyme that they are producing this hydrolases enzyme in order to produce these herbicides, which are being used in the farmlands to kill the herbs. So with the help of S moipa, and at the end what they're getting with this N and this 1 2 3 4 carbon chain and oxygen here present adding to this group to produce this herbicide. So products that are produced from the industries are carbohydrates, majority is carbohydrates, peptides also is in the majority, and amino acids and secondary alcohols. So and beyond that some chiral compounds also. So if we see in this chart, the major four parts that are playing role are Secondary alcohols, amino acids, peptides, and carbohydrates are the highly recommended part from the industrial perspective. Then comes your fat derivatives, steroids, non-chiral compounds, nucleotides. And but that's how this pie chart is showing us the products differentiation of various products. So I keep checking if you have any questions, students. Though we have a small group, I wanted to do these classes over the Zoom or Google Meet, but I cannot actually record them. In order to do so, I kept these YouTube streaming things open for all of us so that we can continue our lectures in easy peasy manner. I hope all things are fine. Please give me some yes or if all is fine so that we can continue further. Otherwise, it's yeah. Please confirm me if all is fine so far by saying yes so that we can continue next. If you have any question, you can ask me now. Purbita, all is fine. Okay, so I continue. Some product markets. What are the products that are being produced with the help of biocatalyst? So mainly, it's the sector that is growing at the moment is the pharma. If you check some big pharma companies, Cipla, Ranbaxy, Biocon, in Baddi, in the northern India, Himachal, there is Baddi. So all the production is done with the help of majorly because of the biocatalyst. Then comes your sector is the food sector. Food products are being used there. Then several other sectors are there. Agrochemical sectors is there. Cosmetics and polymers are available in that. So this was given by Strathoff, Panker, Schmidt in 2002. The production of fine chemicals by biotransformation. And what is biotransformations? What enzymes are used as a catalyst? So biotransformation is just to produce some new products with the help of microbes. Basically, your biocatalyst enzymes, microbes that we can use. So in that case, various enzymes roles are being shown in this pie chart. That is, hydrolases are being used um, from 65 to 45%. Lysases is around 12%, isomerase is around 4. Reducing cells and oxidizing cells also having the largest use there, around 28%. Oxidative very less, but transferase is still around 11%. So majorly that enzymes that we use are from the hydrolases perspective and also from the your oxidative perspective. And then beyond that, lysases and transferases play the role, but then isomerases and oxidatives are the least used enzymes in the research. So this was shown by Strathoff, Punkin, Schmidt also in 2002. You can watch this paper in the current opinion of biotechnology. So this is a paper showing from Nature Biotechnology in 2004. Difference between your chemoautotropes and photoautotropes. There are four different perspective: chemoautotrope and photoautotrope. how the things are working from the genome analysis of your Rhodopseudomonas polystris. So here we can see on the left, we have our carbon dioxide coming from outside into this chemoautotroph. Like, chemoautotroph means they're producing their food with the help of chemicals. Chemoautotroph. Photoautotrophs, they're producing their food, their source of energy with the help of light. Photo means light here. So in this chemoautotroph, we can see that it's taking carbon dioxide from outside and hydrogen atoms from outside, and also thiosulfate is getting in, and they are producing CH2O, and then they're producing energy in the form of ATP. So that's how, with the help of all these chemicals, our chemoautotrophs are producing things, and they are in exchange removing some extra hydrogen outside, and also water molecules are being produced. And another form of chemoautotroph is shown is here by lignin monomer organics. So we have lignin monomers being added, and they are producing ATP in order to produce energy, and CH2 and the further reactions are happening. Whereas in the photoautotrophs, carbon dioxide is there, thiosulfate is there, everything is there, but it's also need light as a form of energy in order to produce ATP at the end. And lignin monomers organics are also being used in order to do so. So types of reactors, in order to increase your, from the fermentation perspective, in order to increase your industrial, increase the enzyme production, we use batch reactors, fermentation reactors, downstream processing bioreactors. So for the same, we have fed batch reactors, unknown reactors, continuous stirred tank reactors, continuous plug flow reactors. So all these are playing a big role here. Then types of biocatalyst in the industrial biotransformation. So we having free enzymes, immobilized cells, free cells. So they have different perspective, different number of processes are being used, but highly that we use in the biocatalyst are the free cells. They are being the highly used. Then comes the one immobilized cells and free enzymes. Then comes your immobilized cells itself. Then the one which are not reported yet. So how your enzyme activity is dependent upon. So we will discuss on that part in deep manner in our upcoming lectures. So enzyme activity is basically dealing with its kinetics, the mechanisms that is being governed in it, molecular dynamics, yeah, that how the level of your enzyme is working out, expression level, various cofactors like pH, redox level, whether it's being inhibited by substrate products or any other another entity into the enzymatic reaction. Glycosylation, phosphorylation are also playing a big role in the enzyme activity. So this is this equilibrium units are given, Michelson-Menten constants so are given here for particular typical parameters. We will discuss about them in details in upcoming lectures. And further, what productivity is needed for synthetic applications, how much productivity we need in order to produce things in a good amount. So industrial bioprocesses, if we see from the biotech process, the enzyme phenylethylamine, phenylethylamine. So we produced around 400 to 1000 gram per liter per hour. So we are producing until, like, around, let's say on average 800 gram of your phenylethylamine per liter of your solution, or in per hour also. And same for acrylamide enzyme 400, acetate only 5 grams, citric acid only 1, and riboflavin only 0.2. So you can see these biotech processes, using various biotech processes, we can produce from the range of 0.2 gram of particular enzyme to the 400 to 1000 gram per liter per hour. So each things matters a lot. It makes a lot of sense that you know how things are working out from lowest to highest range. And the chemical processes, some heterogeneous catalysis like acryl, acrylonitrile, methanol, amino group. So all these are being also used very well. Around from 10 gram, methanol is around 500, 2000, and amino acid from 1000 to 4000. So how good we have to be? So annual production is over 1 ton in each case, and 1 to 4 processes evaluated. So we can see compound class like amino acids, alcohols, carbohydrates, beta lactams. So this is a very nice summary is being shown here. Epoxides, amines, amides, and various enzymes that are being used in those categories, in those classes, that how much productivity is done volumetric, and then how much we get the final concentrations in gram per liter, and then what is the yield that we are getting there. So if we say in the oxygenase the volume is 1, then the final product concentration will be 7, and the yield is around 90% out of it. Yeah. And here we can see that in this decarboxylase, oxireductase, amidase, lysase, so 54.6 gram per liter per hour being produced, and the final concentration is around 102, and the yield is around 82%. So all these things matter. So here we can see 42 oxireductase and hydratase, and then the final product is 225. You can see from 42 to 225 the final concentration, and the yield is around 96% at the end. So this is how annual production is done about these various enzymes. So now um, so I just take 5 minutes break. Yeah, students, you can also take five. It's 8:20, so we meet at 8:25, and then we start again. Yeah, so I just need 5 minutes break because I'm speaking from 7 a.m. in the morning, so need little bit break now. So I will come in 5 minutes, and then we start again. So welcome back, students. So we continue from here onwards, fundamentals of protein chemistry. So these are the various the amino acids that are playing role, and how these amino acids are making sense. It's from the glycine. These are the non-polar compounds: alanine, valine. The main group of amino acids, that you carbon amino acids, carboxyl group and H group, they will remain constant always. You can see in the glycine, in the alanine, in the valine, mainly your methyl group, your three groups are present: alanine, valine, glycine. Then the leucine is present here, then the methionine is present here, and then the isoleucine is present here. So all these things matters a lot, like this. Then comes your polar particles. Polar means in these we don't have OH group present at the end, right? But in them we have OH group, and also they are more like uncharged R groups are present. So like serine, threonine, cysteine, proline, asparagine, and glutamine. So at the end we have positively charged R group, negatively charged R group, aromatic R groups. So that's it. All these things matters a lot. So we are done. We have discussed quite in a good amount about basics of enzyme engineering today. So this is not required, or I will share with you all these things later. So I keep them. So these are from my colleges I made. So that we have discussed today so far about the historical perspective and enzyme classification. So now we went to some more core part. of it, that is how the enzyme action works and how what is the mechanism behind it. Yeah, so we will discuss mainly this thing in our next 30 minutes of lecture. So let's start with this. So enzyme mechanism and action. So to familiarize, so in this part we will study about various biocatalysts, how biocatalysts are working, to study their types and their structures, and to study the mechanism of enzyme actions and what is the specificity of enzymes and its function. So define enzymes. So enzyme act as a biological catalyst. Um, so they are the proteins that increase the rate of reactions by lowering the energy of activation, and they catalyze nearly all chemical reactions taking place in cells of the body, and they are not altered or consumed during the reactions, and we can reuse them again and again. So that's the basic part, enzyme as a biological catalyst. Some important terms in order to understand the biochemical nature of an activity of enzyme. First is the active site. So active site is the area on the enzymes where the substrate or a substrate attached is called as active site. So we all have these enzymes on active site where substrates get attached and make it to workable. So enzymes are usually quite big proteins. So these questions being asked in some exam also, that what are the nature of enzyme? Basically, they are not carbohydrate, lipids. Basically, they are proteins which are having active sites, a very small region for the substrate. So this is how the active site look like. Enzyme molecules, they contain a special pockets or clefts called active sites here. So you can see we are having this active site here and we having a substrate. Yeah. So substrate is entering the active site of the enzyme. Okay. And then after it's being entered, the substrate binds to it and slightly enzyme changes the shape. So this is your first enzyme substrate complex is made. So this is your first enzyme substrate complex is made. Then the enzyme product complex is being produced. After the production of enzyme product complex, the production of आपके products produce will be done in this form. So this is a very unstable state in between. These two states are very unstable. Once these states are over, it produces our products leaving the active enzyme sites. And this enzyme could be used million times within 1 minute. So lot of substrate could come and leave the product, lot of substrate could come. In between, it will make enzyme substrate complex, enzyme product complex, which is quite temporary, and then afterwards your products are being produced. So that's the basic function of your enzyme. Is basically that's the key role that you have to look for. Then substrate, the reactant in biochemical reactions is termed as substrates, and when substrate binds to enzyme, it forms an enzyme substrate complex. So as we have discussed, what is substrate is, whenever the substrate will attach to the enzyme, it will join together. So it will make a enzyme substrate complex. So I'm teaching all these things first because even though they are basics, but they will help you to understand the kinetics then later. So in this enzymatic reaction, the substance at the beginning of the process on which enzyme begins, it's called action is called substrate. Now comes your lock and key model, as we have discussed just before, discovered by Koshland, that in the lock and key model of enzyme action, first you have your key substrate, then you have a lock enzyme. Then we correctly fit and it will react to that. If it is incorrect, then there will be no reaction. So in the lock and key model of enzyme action, the active site has a rigid shape. So it will shape to the lock. So only substrate with the matching shape can fit. So only the key which is shaping to the lock will fit it here. So our key is substrate here, lock is enzyme. So if it is fit, then it will react. If it is incorrect, there is no, in this like this, the upper substrate we having in this key one joint here, but in this not, so no reaction. And the substrate is a key that fits the lock of the active site. So substrate is the key only which will fit to the active site and make it active and in order to produce the product. So this explains that how specific your enzyme is. So this explains the specificity of the enzymes, and this also explains the loss of activity when the enzymes denatures. So whenever the enzymes here, the log is not working anymore, it's also loses activity then later on when it's denaturing. Then further, this is very common. MCQs are being asked in exams. Apoenzyme and holoenzyme. The enzyme without its non-protein portion is termed as apoenzyme and it is inactive. So the enzyme is known without its non-protein and it's in an inactive state, that will be known as your apoenzyme and it will be in inactive state. Whereas the holoenzyme is the one which is an active enzyme with its non-protein component. Holoenzyme is an active enzyme with its non-protein component. So here we can see in this apoenzyme we are inactivated and we have this cofactor which is an activator and which is also a non-protein portion. And this coenzyme will attach to this apoenzyme and make a holoenzyme. So ACH, you can remember this as ACH. A plus C give rise to H. When it is initially, this is your inactive, you have your activator attached to the coenzyme, and afterwards you have your holoenzyme attaching to the with the cofactor, making it an active enzyme. So once it is active, then only the substrate could come and could attach to it, and your reaction will be good to go further. So remember these part that A plus C give rise to H. Remember always this thing. Then comes cofactor. That is, cofactor is a non-protein chemical. So a cofactor, like we have discussed here, what is this cofactor is this coenzyme. This is nothing but a non-protein chemical compound which is bound in enzyme and is required for catalysis. So this is a non-protein chemical which is act like an activator which attaches tightly or loosely to the enzyme in order to do the catalysis. So there are two types of cofactors, that is coenzymes and prosthetic groups. Coenzymes are the one which loosely bound and prosthetic groups are the one which tightly bounds. So the example of coenzymes are vitamins, compound derived for vitamins, and these coenzymes are non-protein components which are loosely bound to the apoenzymes by non-covalent bond. Whereas the prosthetic groups are the non-protein component tightly bound to the apoenzyme by covalent bond and is called as prosthetic group. Yeah. So that's the main difference between your coenzyme and prosthetic group. That is, your coenzyme is mainly bonded to the apoenzyme by non-covalent bonds, whereas the prosthetic groups are the non-protein components tightly bound to the apoenzyme by covalent bond and is called as prosthetic groups. Then enzyme specificity. So enzymes have varying degrees of specificity for substrates. So enzymes may recognize and catalyze a single substrate, a group of similar substrates, and particular type of bond. So again I will repeat about enzyme specificity here, that they have varying degrees of specificity for substrates and they recognize and catalyzed, and they have single substrate, a group of similar substrates, and particular type of bond also. Then comes your activation energy, energy of activations. So basically your enzymes, it increases the activation energy and make your things to push to the start reactions. So all chemical reactions, they require some amount of energy, some amount of level in order to get them started. And then when they got the first push to start the reaction, that energy is called as activation energy. So here we can see without enzyme, if we having this lactose, and this is these are the bonds that are being broken, then you have glucose and galactose. So this is the initial reaction is required in order to break these bonds. without enzyme, and the net energy released after splitting of the lactose here. So this much is the total amount of energy being released after the splitting of lactose or making glucose and galactose, and this reaction is done without enzyme. But in the same case, if we add enzyme, so our activation energy will be reduced. It will be this much only in the case of enzyme. This is with enzyme. Okay, so keep this thing in the point note, yeah. So that mainly your what is enzyme is doing is just reducing the activation energy of your reaction in order to make things work out properly for you. So let's go next. So this is a summary of what we have studied so far, that enzymes are divided into two parts: simple only protein, or complex holoenzyme proteins, and non-protein factors. Then apoenzyme protein part, it includes your cofactors and apoenzyme, and the cofactors includes your prosthetic groups and coenzyme. The prosthetic groups includes small inorganic molecules or atoms, usually tightly bound to the apoenzyme, whereas the coenzymes are the large organic molecules which are loosely bound to apoenzyme. So mechanism of action of enzymes. So an enzyme increases reaction rates by decreasing the activation energy. So whatever that we have discussed in the previous figures, so it decreases the activation energy to this much level, so that we can have better results. So mechanism of action of enzymes. So enzymes increases rate of reaction by decreasing the activation energy. Also there is enzyme substrate interactions in which formation of enzyme substrate complex is done initially. And then lock and key model is also there, and induced fit model is also there. So all these things are the coming under the mechanism of the action of enzymes. So we discuss now what is the basics behind this enzyme catalyst reactions, how the enzyme catalyst reactions are working out. So when a substrate fits properly in an active site, so enzyme substrate complex is formed. So ES, like we have discussed in our first image today, how E makes ES, then ES makes EP, then EP makes E plus P. That is enzyme and substrate, they come together, and then the enzyme substrate complex is made, then enzyme product complex is made, then you have your enzyme and product at the end. So E plus S giving rise to ES. So within the active site of ES complex, the reaction occurs to convert substrate to product. ES give rise to E plus P. And the products which are released allow another substrate to bind the enzyme. So another substrate could attach, and the millions of cycles are possible per minute. So many substrate could be attached to enzyme, and they keep producing millions of products also within a minute. So the overall reaction of the conversions of substrate to product can be written as this way. So E plus S give rise to ES, which is a reversible reaction, and then ES gives the irreversible reaction giving rise to enzyme plus product. Then enzyme substrate complex, we can see enzyme and substrates combine to form complexes, that is E plus S giving a reversible reaction. That's the step 1, it's make a complex. Then in the step 2 we have a enzyme substrate complex is formed in the transition state, and then giving rise to E plus enzyme plus product state. And then this enzyme and product, they got separated, EP giving rise to your products are being made, and enzyme is again another substrate here. So the outcome from today's lecture that we have studied is that it's provide a complete detail of enzyme. It introduces enzyme as a biocatalyst and explain the concept mechanism of enzyme action here. So on that note, I'm ready to take your questions. So I will share these presentations with you now. I will share you afterwards. Yeah. So if you have, so we have just discussed on the first day of our lectures, historical perspective, enzyme classification. We have discussed enzyme substrate interaction, ES complex, binding site and active sites. So we are covered with the A part and E part for the today's lecture. And I want to continue further a little bit about nomenclature also, classification, how the kinetics works for the same. So that we will do in our upcoming lectures. So I would like to make a pause here, and then and I will also share one book with you. It's actually Lehinger book. So if you want to study about this topic, about enzyme thing, I will highly recommend to go to chapter number 6. Yeah. So all details that we are discussing, what we have discussed today, it's been given in these chapters in a very easy and nice manner. So chapter number 6 from Lehinger book. So I will share this PDF with all of you now in the group. So chapter number 6 from this. And I will share the today presentations with all of you, whatever we have discussed just now, so that we can continue with that. Any questions, students, so far? I am open to your questions now. Student, I need your reply. All is fine so far? Because I cannot start the another topic because we have only 10 minutes left. If I start that topic, that will be left in between. In between, in your meantime, we will see us tomorrow again. You can go through all these chapters in order to understand the depth of enzymology, enzyme engineering actually. So all the concept that we have discussed, they are given here. So we will discuss soon about competitive inhibition, uncompetitive inhibition, and mixed inhibition. So we will discuss how these reactions are playing role here, and various reactions also. Okay, so that we might finish it today earlier, and then we see us again uh, tomorrow at 7 a.m. again, and we will continue with the same lectures again. Continue with the topics, with the classification and how the main things are working for the same also. ठीक है class. We will finish. We see us tomorrow then. Thank you very much. Take care then.